For about 65,000 years, my hometown of Waco, Texas, has harbored a great big secret. And that secret is really a story, and that story is really a tragedy. In the late 1970s, a couple of young men were hunting for arrowheads and spear points down by the confluence of the Bosque and the Brazos rivers, just below the Lake Waco Dam, if you're familiar. And instead, they came across the femur of a Columbian mammoth that had been dead for about 65,000 years. So about 40 years ago, Waco's big secret was literally uncovered. And interestingly, most of that story is still under wraps and plaster casts in a museum at Baylor University waiting to be interpreted. By 1990, the fossil remains of 16 Columbian mammoths were discovered at this site, which is just located a few miles from my childhood home. And it seems that a nursery herd meaning mama mammoths and baby mammoths, had died together in a single natural calamity. During the 1990s, 6 additional mammoths were excavated, including a large male, and crews have also uncovered the remains of a western camel, a dwarf antelope, an American alligator, a giant tortoise, and the tooth of a juvenile saber-toothed cat, and 4 Budweiser beer cans from my high school graduation party. Based on these finds, the Waco Mammoth National Monument became a reality about 6 years ago. Our guest today is Dr. Lindsey Yann, and she is the paleontologist at the Waco Mammoth National Monument. Dr. Yann completed her undergraduate work at Virginia Tech, collecting dual degrees in geological sciences and in biological sciences, magna cum laude. And in addition, she earned her master's degree in geology and geophysics at Louisiana State University. And Dr. Yann completed her studies with a PhD at Vanderbilt University. She's published more than a half a dozen peer-reviewed papers. One is entitled Global Long-Term Stability of Individual Dietary Specialization in Herbivorous Mammals. I know y'all don't care, but I just wanted to read that out loud. Dr. Yann is a teacher and a lecturer at the university level at several well-known schools. She's led fieldwork at numerous fossil sites across the country, and I saw her quoted in National Geographic magazine earlier this year. How about that? So with that, it is my pleasure to turn it all over to our guest speaker, Dr. Lindsey Yann. You folks are in for a treat.
Thank you so much for that introduction, and I'm so happy to be here with you guys today. As I share my screen, I'm going to go ahead and turn my camera off. Actually looking at is a reconstruction of what we think that Waco might have looked like during the Pleistocene. So what I'm going to talk about today is a little bit of how we can interpret diet and climate using these fossil animals. What I'm going to do is I'm going to go over some of the major climate and environmental proxies that we use in both modern and paleontological studies. And then I'm going to provide you guys with a couple of ways that we interpret the fossil record. And then I'll use some of these to give you some actual research that I did for my dissertation, and then wrap it up with a look at Waco Mammoth National Monument in case you guys haven't had a chance to come visit. So the first thing is when we reconstruct environments today, the most common way that we do this is using historical records. So hundreds of years of data from all over the world, and we can use that to see trends in how climate, precipitation, and temperature has changed. Or we can look at coral in the ocean to get a better idea of what the ocean itself is doing. We look at modern plants and animals, and we can see how their ranges have changed or how they have adapted or evolved with the different climates and different environments. Commonly, you hear about ice core records. They're one of the best ways to get recent past, but thousands of years into the past. With that comes water records and all of the organisms living in the water, like our corals, but we also use these little tiny things called forams, and I've got a picture of them on the next slide. Another one of our really common ways is looking at tree rings. So you can get an annual record of what a good growing season or a bad growing season might have been in these tree rings. And then same thing, we can see these layers in speleothems, or these are cave deposits. So stalactites and stalagmites. researchers will cut them open and look at them, and they have tree rings pretty much identical to an actual tree. And you can go through and track different climate using that rock record. But a lot of times these more modern proxies aren't around for us to look in the fossil record. So we have to rely on different proxies or different ways at looking at the fossil record. And some of the really common ones that we use today are these little ostracods up here. These are basically shelled shrimp, and they're really good at tracking water environments and can be a good environmental indicator. These are the foraminifera that we see a lot in ocean sediments, and they've actually been used to track changing temperature for the last 100 and something million years. So this tiny organism, which is smaller than the size of a pinhead, can give us all of this information about climate. We've got dinoflagellates. We've got these here in the middle. These are phytoliths, or little bits of silica, or kind of like a teeny tiny grain of sand incorporated into the plant tissue of a grass in this situation. And those tiny little pieces of sand grains, they can wear on tooth enamel, and they can tell us all about diet, which we can then go through and identify environments off of that. We've got pollen grains, which are great for reconstructing the floral history of an area. And it turns out that they preserve pretty well, and they are very similar to pollen grains that we have today. So it gives us a chance to look at the plant record. In addition to the pollen, we also find charcoal fragments in historic and prehistoric sites, which allow us to, again, better understand how frequent fires occur and how that might play a role in how we interpret the, especially the aridity of an area. And then some of the neat things in more modern research is looking at some of the chemical signatures of plants and animals within the rock record or within the soil record. So if we're able to find something like chlorophyll A, this compound in the soil, we can then begin to get a better idea of, again, what the environment looked like. So even though we might not have that pollen grain or that little bit of charcoal, sometimes we get lucky and we get those chemicals which can be analyzed, and they can give us additional information that maybe we wouldn't be able to get at the site otherwise. So these are some of the really common microscopic things. So these are all, you need high-powered magnification, and you need expensive equipment. And this is not something that I've spent a lot of my time on, but it is something that is crucial within paleoecological and paleoclimatological studies. But I'm going to go into more of the macroscopic. So this is more along the line of what I've worked with. So things you can see with your eyes. You don't need any special equipment. The one over here, this is a sediment core. And if you look really closely, there's a space here where you change from really fine-grained sediments to a little bit coarser sediments, and then you've got a bunch of shells here. So this core of sediment is thousands of years old, but we can still go through and look at specific small pieces within that, and we can interpret what the sediment was like, maybe the depositional environment. So what did the whole basin look like while these animals were living? My personal favorite is using vertebrate remains or bones and teeth from animals. We can look at the overall morphology or shape of a skull or a long bone or a tooth. We can also go out into the field and we can actually watch animals eat and interact with their environment. So like this African elephant here, we don't have to guess at what it's eating. We can literally go out and watch it eat, which really allows us to pinpoint the specifics of what they eat and then how they can change their diet as they need with modern changes to the environment. One of the other common proxies are using clamshells or other bivalves. Turns out that when these animals secrete their shell, they incorporate the geochemistry of the surrounding water. So by looking at the annual growth rings in the shell, you can go through and see changes in time. And then same with things like coral. And some of these can be extremely large and extremely old. So even if the original coral in the heart of this blob has died, which it has because it's cut off from the outside, a scientist could go in and look at the original record within that blob and get a history of how that spot's changed. And then we also can use fossilized plant material or modern plant material, biological surveys to go out and just investigate like what plants are common in this national park. And that can serve as a basis for future studies. This is where I'm going to start having you guys do a little bit of thinking, and then a couple of slides in, I want you to put your answers to the questions that I ask in the chat, and Jean or Chuck will go through some of those answers so that you guys can get a little bit more interaction. But the first thing that I really want to know is, well, how do we go from a fossil to a diet? So I can look at a fossil animal and I can look at things like its overall shape. So this is the morphology that we were talking about. And that's probably one of the most common methods for inferring diet from bones, is looking at the overall shape of them. So we're going to go through these three organisms that we have on the screen here. So the first one, we want to look at the skull. So the first thing we look at is its teeth. We've got its molars or its cheek teeth here, and we've got our incisors here. And we notice there are no big sharp canines, and there's this really big gap with no teeth here, or a diastema. Other things we look at are the eye orbits, and are the eyes pointing to the side, or are they pointing more up front? We also look at the jaw joint. So here's our tooth row, and then the joint of the jaw is way up here. So it's much higher than the teeth. We also look at the overall jaw shape and the length of the skull, specifically looking at the nasal bones here, so in the nose. And what we see is with this one, the eyes are pointed fairly outward. The jaw joint, again, is well above the teeth. Our jaw is curved, so if you actually were to push on the front end of this jaw, it would rock back and forth. And this is considered to be really elongated nasals. So this skull is a white-tailed deer. We can go out in the field and we can watch it browse on trees and shrubs. So these are some of the features that we might expect to see in an herbivore. If we move on to the next skull, we look at the teeth first again, and we see we've got nice big canines. We've got some smaller peg-like teeth back here. And then we've got these wonderful meat shearing teeth called the carnassial. And these are pretty much like scissors when it comes to meat. If we look at our eyes, we've got an eye that's pointing more forward than in our last specimen. Jaw shape, we can see there's a little bit of a curve down here, but not nearly as much as we saw in that deer. And then if we look at the nasal bones, it is elongated, but not to the same extent that we see in the deer. So it turns out that this skull is a coyote, and these animals are omnivores. So they're not going to have the same morphology that we saw in the deer. They're going to be somewhere between the morphology of a carnivore and an herbivore. And oh, I guess I should point out too, the jaw joint here, This is pretty much in line with the tooth row, which allows for more biting and tearing instead of the side-to-side chewing that we see in the herbivores. So if we move on to the last skull in this bunch, we see here, again, our nice big canines, our peg-like teeth here. And it's harder to see in the way that this is oriented, but these are some really nice shearing carnassial teeth. We've got eyes that are pointed nearly directly forward. Our jaw joint is completely in line with the tooth row. We've got a completely flat jaw, and we have very squished nasals. So this is actually a North American river otter, and it's a hypercarnivore feeding on mainly fish, but pretty much any other vertebrate or even invertebrate that it can get its hands on. So this is that true carnivore shape of a short skull, flat jaw, and sharp teeth. So here we've got to look at their morphology. Now I want to show you a few other options, and this is where I need you guys to chime in. What do you guys think that this animal eats? So we're got to remember, examine the eyes. How are they pointing? Well, I guess start with your teeth. What do you see in the teeth? And then look at the eyes, the jaw joint, which is here. It's a little bit above the tooth row. Got the jaw shapes fairly flat and a moderately extended nose. So does anybody have any guesses on what this animal might eat?
You guys put it in the chat and I'll track them for you. Well, here we go. Chuck says carnivore. Mike Valdez says a carnivore. Who else? I missed another one up there. Meat. Somebody says they eat meat. Omnivore. Pam says an omnivore.
Okay.
Another omnivore, meat eater, carnivore, meat. On and
okay,
on.
So it seems to be a consensus on it's either an omnivore or a carnivore. Okay, now I want you to try with this picture. What do you think this animal eats? Again, use these examples or these features over here to make some estimations. One thing I want to point out here is look at that jaw joint. It's a whole lot higher than that tooth row.
I'm looking. I can't tell if Mike was guessing on the last one at omnivore and it's Cindy. Here we go. God, it goes so fast. This is a bad idea. Who was thinking when they did this? Anyway, omnivore. How about that? That's what I'm getting. Omnivore.
Omnivore. Okay. And then this is the last of the skulls in this batch. What do you guys think about this one?
Not seeing anything yet. Plant eater was Pam's last entry. Mostly leaves, Skip said earlier. Plant eater, carnivore. Two different things. Omnivore from Coral.
Okay.
I think we're guessing.
So the next questions are, what animals are these? Can I have some guesses on A? What do you guys think that this one might be?
Canine. Michael says canine. It looks like a dog. I'll give you that. Dog. Pam says dog.
All right. What about B?
It's my Uncle Bob. I'm with a primate. Leanne says primate. Chimp. Gorilla. I think we landed on something there. Yep.
And what about C?
No guesses. Cat. Chimp. Yeah, I'm sorry. I'm reading an old one. Cat for sure. Cat. Big rat.
Ooh, big rat. All right. So it turns out that this one down here, this is a fruit bat. Its skull is only about 2 inches long, and it only eats fruit. This skull, you guys were right, it is a primate, but it's a howler monkey. And these animals pretty much exclusively eat tree leaves. And then this one over here is the giant panda, which only eats bamboo.
Well,
So
Just know
only
this.
go off of, what was
Know
that?
this. I was gonna say, Joe Bryant, he guessed a bear, so he's close, right? Bear's
There we
like a
go.
panda. Okay. Just to give Joe credit.
Good job, Joe. But what it is the typical features that we use when we're talking about morphology can sometimes throw us off. So morphology alone really isn't enough to understand the fossil record, because with all the specimens on the screen right now, somebody went out in the field and watched these animals eat. So they know that even though this bat has crazy sharp canines, it only is using those canines on fruit. But if we just had that skull in the fossil record, it would be really hard to make that interpretation. So if we're not able to look at skull morphology, then what else can we do? And it basically comes down to a combination of using morphology and other proxies. So we use the modern analogs, we use the morphology, we use what we know about an elephant, and we can use that to try and make some interpretations of what a mammoth was doing. So we can't go out into the field and watch a mammoth, but we can watch its next closest relative and use those behaviors and that morphology to get a better understanding. We can also use other types of remains. Anyone want to take a guess to what this is?
Coprolite, poop, poo, dung.
Yes. So this is a coprolite. This is from a giant ground sloth. It was preserved in a cave in Grand Canyon. We can also use microscopic features like this. This is a 3D representation of the microscopic surface of a tooth, and we can use that to help us infer diet. And then we can also use geochemistry. So this is my personal favorite, and we'll talk more about this in a minute. But I'm really interested in how the geochemistry within an animal is incorporated into its tissues and what we can then learn from that. So if we look at our modern analogs, up here at Waco Mammoth, our elephants and their relatives or proboscideans are really one of the major focuses of what goes on up here. So we can look at the teeth that we have in our dig shelter here. So this is an upper tooth from one of our females, and we can look at its shape. We can then look at the African and the Asian elephant tooth and realize that our tooth looks a lot more like this Asian tooth. So we then can go and look at the Asian elephant instead of the African elephant to really get maybe a clearer picture of what they would have been doing during their life. Some of the other things that we can do with modern analogs is we see things like this. So this is a bone that's in our dig shelter. It should be a nice straight rib, but there's a softball-sized break. And then another one in our collections at the Mayborn Museum at Baylor University. This again should be a nice straight bone. And we see this really elongated and thickened area. So these two are very common breaks in modern elephants. So it turns out that this is a break from when males fight and they take their tusks and bash them into each other and they break ribs. And then this is a common one on females, and it's a result of mating injuries. So we're able to watch these modern elephants interact and interpret the fossil record. And if we look a little bit more at some of the fossils that we have here in Waco, we've got a skull. And by looking at the teeth and the overall shape, we're able to determine that it's a camel, and it's most closely related to our dromedary and Bactrian camels. So we know the kinds of foods and the behaviors they have. We've got these bones here, and this is part of a lower jaw from an alligator. So we know we had alligators, which tells us that we had water. In order to support an alligator, we couldn't just have a tiny little mud hole. We had to have substantial water. We also have some of these bones that haven't even been really prepped out yet, but that is a leg bone. And then this is a paw that's fully in the life position, still in the rock. It's hard to see, but you can see individual finger or toe bones there. And they come from a coyote. So even though we don't have many fossils of carnivores, we're still able to identify the fact that we have that coyote, and we understand how that animal works within the ecosystem. So we can interpret the site here a little bit better. And some of the other proxies, I already mentioned this a little bit in the first slide, we can use dung. So we've got this 100,000-year-old ground sloth dung, but it's not the only thing that we find preserved in the fossil record. Here is another form of ground sloth dung. This is a patty from a mammoth, a Columbian mammoth to be specific. So it was found in association with other bones. So we know that all over the landscape here in Waco, we would have had these giant patties of mammoth dung. And sometimes, especially in Arctic environments, you get even better preservation. So this crazy looking thing up here is a mummified bison. And it turns out that within the center cavity of this bison, the stomach contents were preserved. So you can go in and dissect this blob and see exactly what plants this animal ate. We can look for pollen. We can look for different chemicals. And you can also look, so this is actually the mummified stomach. So you can look on the inside of the stomach for those chemicals as well. So not only can we rely on the hard skeletal items of an animal, but every once in a while we luck out and we get some of these more soft tissue preservation that can tell us a lot about the very specifics of what these animals are eating. Because when I talked about the phytoliths, or those teeny tiny sand grains, they often survive through the stomach of an animal. So we can look through this pile of dung and identify what animals were on the landscape. And there's really not another way to do that. So unfortunately here at Waco, we have not found any dung, so we're not as lucky as some of these other sites, but we'll keep looking. This is a crazy representation of the microscopic wear on a tooth. So all these tiny little lines here are made from those individual sand grains. So as a scientist, you go in with a confocal white light microscope. And what you're looking is, you want to know, like, what orientation did this animal chew? Or do we see all of the scratches go in a single direction? How rough was the surface? Was that animal eating really hard seeds, or maybe it was crunching bone? So if we found an animal that was just eating meat, like a cheetah, we would expect nice, fine, linear features. But if you think about a hyena that's just crunching bone and eating all sorts of things, we start to get into these really complex patterns. And the result are things like this, where you've got a nice orientation. And then this one at the bottom, it's kind of just all over the place. You've got scratches and you've got pits. So we can use the differences in this wear to begin to infer diet. So one example of this being used in a recent paper looks at a mammoth, mastodon, and gomphothere material from Florida and Texas. So what we're doing is, these are the different teeth. So this is Mammuthus, oh my goodness, tripping all over my words. So E is the Columbian mammoth, D is the Cuvironeus, or the gomphothere, and C is our mastodon. So mastodons have been interpreted as eating browse or trees and leaves. And we can kind of see that there's no real nice, pretty pattern to the mastodon. Our gomphothere, you see these lines intersecting, and you see some high points here and some lower points. But then you come over here to the last one, which is the Columbian mammoth, and you can see these very obvious, almost left to right lines with an occasional different feature. Well, it turns out that this is what it looks like if you eat really abrasive grasses. And this is what it looks like if you eat those nice soft leaves. And this has been ground truth by doing direct feeding studies of different animals. So they will feed them grass with added dirt, or they'll feed them leaves with added dirt, and they can go through and quantify how different quantities of dust or debris really impacts the tooth wear. And then once you know what the animal's eating, then you can continue on to that environmental interpretation. Now we're going to my favorite part. So most people really don't care about stable isotopes, and I don't really blame them, but it turns out that it's a wonderful technique for looking at diet and climate, because you are what you eat and drink. So if we start, what is a stable isotope? So most people have heard of carbon-14 or radiocarbon dating. That's a radioactive isotope that breaks down. I'm not interested in those for this study. What I'm interested in carbon-12 and 13. And the difference between these is carbon-13 has one extra neutron in the nucleus. So it's not any different charge, but you've got that extra blob in it, making it just a little bit bigger and a little bit heavier than the carbon-12. And I will show you why this becomes important. But I'm also interested in oxygen. And here we're looking at water. So you can have oxygen 16 or oxygen 18. And again, this 18 is a little bit bigger and a little bit heavier than the 16, and we can use that to make some interpretations. So the first thing that we have to look at is photosynthesis. So basically the way that plants take in carbon dioxide, or for my case, the actual carbon, varies based on the photosynthetic pathway. And I'm not going to talk about CAM plants because they are not within the study area that I looked at, but they would kind of fall between a C3 and a C4 plant. But our C3 plants, these are our cool season grasses, trees, and shrubs. And then C4 plants are typically grass, including things like corn, but there are a few rare shrubs out there like saltbush that are C4. And if we look at how this works and how we can use it for isotopic studies, this cloud up here represents the ratio of carbon-12 to carbon-13 in our atmosphere. And then we've got a diagram down here of how CO2 is brought into the leaf through the stomata. And then your byproduct is water and oxygen. So if we look at this, the key limiting factor typically becomes water. So the cool season plants, which are C3 plants, these take in the carbon dioxide and convert it into a 3-carbon chain. And the C4 plants, again, create a 4-carbon chain. So if we look at what that means, if our ratio here, this C3 redbud leaf, it brings in very few carbon-13. And the reason is because C3 plants are typically found in habitats that aren't limited by water. So they can actually leave their stomata open for quite a while, and they can go through and they can choose to only bring in the smaller carbon-12. So it's easier to bring in and it's easier to process the carbon-12 than it is 13. So if you don't care about how much water you lose, that strategy works wonderful. But if you're living in an environment where water is limited, water loss is going to cause severe problems with the overall growth and reproduction of that plant. So C4 plants take a completely different approach, and instead they basically open their stomata and take in a big gulp of air. So that gulp tends to reflect the actual ratio in the atmosphere. So C4 plants, you're gonna get your atmospheric ratio, and then C3 plants give you a completely different. So it's these fundamental differences in how a plant decides to bring in its carbon that we can use to tell us what these animals ate. So how do we go from plants to fossils to diet? And that is through lab work. So what I did is I took a Dremel drill, or a small rotary drill with a very tiny bit, and this is a llama tooth. And I was able to go through, and each one of these lines is an individual sample that I took. Same with the lines here on A, and then another sampling strategy on B and C. And then through a chemical process, you end up with a really clean white powder that then gets sent off and analyzed. And we get values back that we then can use to make the interpretations. So with carbon isotopes, you are what you eat, plus or minus a little bit. So if we look at an animal's tooth enamel using that chemical process that I mentioned, if we get values that are near 0, that suggests that that animal was eating just C4 plants. If the animal has a value of -10, that suggests we're eating just C3 plants. And then if you fall in the middle, basically it's a mixed feeder. So they're eating whatever's in the environment. So you can use that information to interpret the ratio of C3 to C4 plants within a particular environment. With oxygen, it's all about your location. So the closer you are to the coast, the closer you are to 0. And that really heavy 18O, it's hard to pick it up, and it's hard to keep it in a rain cloud. So as you move inland and you move up, all of that heavy 18O falls out, and you're left with this really negative rainwater or snow. The other thing that we can see from oxygen isotopes is the difference of aridity. So we would expect lower oxygen isotope values in areas that are very humid, and high oxygen values in areas that are very arid. And one of the places where this has been demonstrated is looking at the tap water from across the country. So we see down here in Texas and Florida, it's basically 0. But as you move up into the Rockies, you get more and more negative. So you're getting further away from the coast, and you're getting higher up into the mountains, creating these negative values. So I used that information to document that camels actually track environmental change. So they track changes in aridity, specifically was what I was looking at. So with that information, I could then start to look at the diet of these different Pleistocene animals and look at how changes in precipitation affected their overall behavior and diet. And this I'm going to show diet, not behavior. So this is some of the stable isotope data from Florida. So in orange is our interglacial site. So we're talking about high sea levels, warm temperatures, and we see this really broad range in oxygen isotope values. And then we've got Inglis 1A here in purple, and this is a known glacial site. So it is during a very low period in sea level, and there are taxa here that are from much cooler environments. So we're able to see that the oxygen isotope values here on the y-axis are much more constrained and lower than our interglacial site here. So if we then add in another site, so this Tribrighton, this is one of the sites I worked on for my dissertation. And it turns out that the values are even more positive than our known interglacial site. So that suggests that Tribrighton was quite hot and likely arid as well, because these values are so much higher than 0. And then if we plot in my second site here, it falls in the middle, and it overlaps with both our glacial and our interglacial site. So this suggests that Hale 8A was probably kind of in between. There's nothing overly drastic about its temperature, hot or cold. Same with aridity. It's kind of just in the middle, probably a pretty nice, equitable climate. And we can also look at what we call serial samples. So the previous, this is all one sample, one animal. This whole row belongs to one animal. So what we did is we took a bunch of samples along that tooth, and it gives us a chance to look at climate or seasonal signals within an individual animal. So if we look here, we don't see a lot of interesting change, but this is our known interglacial site. And we see this wavy pattern, and this matches up very nicely with the modern climatic profile for that part of Florida. So it looks like we've got these nice seasons. So it turns out the winter had very distinct, or the glacial period had very distinct seasons. And then this one seems to be kind of all over the place. And like I said, we can match that directly to the modern rainfall. And we know that these really low points are actually due to the fact that they get so much rain. So you can wash out a particular signal by overpowering it with precipitation. So that's what we've seen here. This is a common humid hot signature, and this is a common kind of cooler, drier signature for Florida. And it turns out that my two sites are completely flat. There is no variability, no significant variability. So it's likely with this one, the animals were migrating because it was so hot and dry. They had to go around Florida to find the resources that they needed. And this one, we still haven't quite figured out exactly why it's flat. But one of the things about where this site is located is there are a lot of sinkholes. So it's probably that this animal was drinking from a sinkhole, and sinkhole water has been mixed over time. So you get this really flat signature. So we can do climate interpretations here with these two profiles, migration potential, and actively, like, what water source are they drinking from? So there's a lot of material can get out of these teeth other than just a single isotope value. The next thing we did, I know this graphic is usually a little bit scary, but I'll walk you through it. These are all individual sites, the same 4 sites, and then each value this way is one individual. And we wanted to know whether they were changing their diet in response to changes in climate, which would be going from glacial to interglacial, or whether they were changing it due to just a strictly floral change, which here at our glacial site, we see C3 plants, and then we see a jump in C4 here. So I'm going to quickly go through a few animals. If we look at our camelids, we've got Paleolama and Hemikenia. They are both found in Texas as well as in Florida. We see an increase in mean values as we cross from interglacial to glacial. And then we don't even see Paleolama until we get into these warmer and hotter sites. We're looking at white-tailed deer here. Again, we see a significant increase in values between glacial and interglacial sites. Granted, we only have one specimen down here, but other taxa here are suggesting that same pattern. If we look at the horse, we see a significant increase here. So at the warmest and the driest site, our animals are eating significantly more C4 vegetation. And the last one, which is more important for our Waco site, is our mammoths. And these, we do see a significant change. We actually see a significant change towards more negative values. So it looks like maybe at these hotter and drier sites, these animals had to use different resources and potentially drink more water. But overall, we were able to use our oxygen values to really characterize the overall climate. And then it appears that that climate from glacial to interglacial is changing the diet of these animals, with the warmer sites being able to support more animals, like supporting 2 camels. We also looked at sites, a site in Texas, in Ingleside, and then when we compared that with a site in California as well. And Ingleside, so this is Texas. The cool thing to point out here, so this box represents the camel values, and we've got 2 individuals over here. And what that tells us is on the coast, we had C4 saltbush. So these animals are known for eating C4 saltbush. And that's the only way that we can figure out to get camels with values this high. So we're able to add another plant to the site interpretation for Ingleside based on these isotope values from a camel. If we look at our site in McKittrick Brea, this is in California, and we notice that the values are all bunched up in the middle. Well, it tells us that this environment is much less diverse than what we see in Florida and Texas. It's very narrow, probably a little bit more arid, and dominated by the C4 grasses instead of the C3 plants that we see in the other parts. So if we put all of these together, it looks really scary, but what we're able to show is that climatic continuum, some wet and cold to hot and dry. So we're able to use these animals to really investigate what the southern half of the country was looking like between 1.6 million years ago and about 50,000 years ago. So basically, we can take chemistry and we can reconstruct that environment. So real quick, I wanted to give you guys a micro tour of the Waco Mammoth National Monument. So this is our beautiful dig shelter. It's climate controlled. So even in the nasty summers, it's only about 75 degrees in here and 50% humidity. We have 2 fossil layers. So down here on the floor of the quarry, we've got a juvenile mammoth, and that is about 65,000 years old. And then we've got 2 feet higher, and we've got more material up here. And these are dated at about 51,000 years ago. So on this level, we have a big bull mammoth, which I can switch. Here we go. We got our big bull. So we got giant tusks, a skull, and body. And there are his hind legs. We also have a female. This is Mammoth W. We can see those nice pretty teeth and her lower jaw. And we've got a camel. So my personal favorite. We've got the skull and some leg bones, and then the backbone going right down there. So we've got more than just mammoths at this site, but we're known for our nursery herd. Like Jean said in the introduction, majority of our site is females and juvenile individuals. But we also have an entire ecosystem. And we can look at some of the other fossils that you can't necessarily see in the dig shelter. So we've got the teeth that we can use for diet. We know that they were grazing grass. This is the kneecap or a patella from one of our mammoths. And you can see these V-shaped features here. These are bite marks. And it turns out that they don't match a saber-toothed cat. They actually match a dog. So likely a coyote or a wolf was eating on our mammoths. We have the presence of a saber-toothed cat. Right now, we just have this juvenile tooth, but we know that they were in the area. And then even outside the dig shelter, we find isolated teeth. So this is another camel tooth. So we're slowly adding to the understanding the grazers, the browsers, and the carnivores at our site. And that's going to become really key to understanding the environment. But we have to look at everything else that's present. And this is something that visitors don't ever get to see. This is all stuff that is housed at the Mayborn Museum. So we have bison, we have horse, deer, rodent. This is a dwarf pronghorn. This is one of our coyote bones. We've got peccary, we've got turtle, fish. So this is a garfish scale. we have drumfish and drumfish teeth. We have our alligator, and then we have a bunch of birds. And nobody's looked at our birds yet, but we have at least 4 different types of birds in the collection. So it turns out that even though most people come here to look at these giant mammoths, we actually have an entire ecosystem. So as we go in to look more specifically at the fossils and their geochemistry and their morphology, we can start to really understand what happened here 65,000 years ago and 51,000 years ago. So that's the last bit of my presentation. So I wanted to open it up and see if you guys had any questions.
Jean, would you like to unmute and ask your question?
Yeah, my question is pretty simple because I wrote it down early on. You mentioned Columbian mammoths. I talk about Columbian mammoths, and I've also heard about woolly mammoths. And I don't even want to get into mastodons, but can you tell us the difference between the woolly and the Columbian?
Yeah, absolutely. So most people, when you say mammoth, they picture a woolly mammoth. So those are the ones that you see in popular culture. They are quite furry, but it turns out that our Columbian mammoths, these are the bigger cousins to the woolly mammoth. So if you look up here, you can see a reconstruction of a mammoth. Well, you only get the butt of it, but it's not furry like the woolly mammoth. It's more, it's closer to an elephant in the amount of hair it has. It's bigger, its tusks are bigger. So basically everything in Texas is bigger. The southern mammoth, our mammoth, is a lot bigger than the woolly mammoth.
And the Columbian, that infers New World, or is that just a word?
It is a New World. It was thought to be a New World when it was named.
Okay, that makes sense.
Been found, I think, in Russia.
Okay, well, that was my question. What I have figured out since I wrote that question is that you could pull my teeth and Chuck's teeth and Janet's teeth and figure out what we ate, what the temperature was, what the aridity, all that can be figured out from teeth and bones, right? That's what you're saying?
Yes, and we use teeth because they're really hard and they don't get altered. It would be really difficult for me to find that out from your bones.
Okay.
But from your teeth, I absolutely could, especially if you grew up somewhere very different than where you live now.
That is just fascinating. Thank you.
Lindsey, I do not see any additional questions, just a lot of wonderful comments on how excellent your presentation was today. And it was so fascinating to watch your manipulation of the data and how you use that to interpret ancient environment. So another question did just pop up.
A couple did.
Okay, Ainsley, would you like to unmute and ask your question? Ainsley?
Just trying to find the button.
What is the easiest distinction between a mastodon and a mammoth?
Mastodons are much smaller, but their teeth are the best way to tell the difference between them. Maybe too far. There we go. Okay, so mammoth teeth, they have these lovely little plates that go this direction. And then a mastodon's tooth has a ring of enamel around the outside. So they've got these loafs or these ridges here. So that's one of the dead giveaways. Even if the bones of the animal look very similar, if you look at their teeth, you can immediately rule out one or the other.
Thank you.
Any other questions? I'm not seeing any other pop up. Other than there is a request that someone would like to see this presentation all over again. That's how fascinating we are with the presentation. And agreed. Well, thank you very much, Lindsey. Like we've said, what a fascinating presentation that you gave today. Thank you so much for spending your time with us.